Thank you, Jim. So I'm going to go fairly rapid fire through some of the evidence here. So we begin with the first slide that you see, which is number 2 in there. Just to give you some context on it, there's been a program for many years in Office of Naval Research dealing with one form of directed energy, that is acoustic energy. It's run by Program Officer Kurt Yankaskis, and some of the work that I'll discuss here, the new work at the end especially, is supported by him in that program. And this is out of Code 34 at the ONR. We really came in when I came in with this with Dr. Hoffer, and he called me about this, I began to think right away, in addition to trying to do some abductive forensics on this, we also tried to think, well, what possibly caused this? The source of the exposure is completely unknown. We've heard before it could include, and I've listed a few of these sources here on the 4th slide, which include hypersonic sound or LRAD, pulsed radio frequency, pulsed laser sources. There are ultrasound devices that can do this, and we're focusing really, the focus we've been talking about here, and Dr. Giordano brought forward, are the receiver characteristics of the human head. What are the waveguide, resonance, and cavitation properties of contents inside a person's head, of intracranial contents? What I'd like to do here now is go through a quick overview of literature, much of it supported by DoD from the 1960s through 1990s on ultrasound and radio frequency effects on inner ear and brain, and point out that organs of hearing include the sacral and utricle. The tests that Dr. Hoffer pointed to, the vestibular-evoked myogenic potentials, actually test the otolith organs, the utricle and sacral, by playing a loud sound into the ear. I'll then show some COTS devices that are available now for ultrasound and pulsed RF emissions to show us that the technology is already out there, and then I'll show some recent findings supported again by Dr. Yankaskis' program on the objective tests of eye movement and pupil coordination that we can distinguish these people in a certain in a field setting. Now, the intracranial waveguide resonance and cavitation work is being done with colleagues at the University of Pittsburgh. Jeff Vipperman and George Klinzing are both professors. Brandon Saltzman and Scott Mang are students here. And directed energy, that is chart 6. If we move to 7, this just introduces the fact that there are a variety of exposures where we already have information on biological effects of directed energy. We can read this in the literature. Chart 8. And what our basic notion here is we're going to characterize the waveguide resonance and cavitation features of the cranial contents. As Dr. Giordano mentioned, blood vessels, in a little more, we go down in a little detail, they're actually surrounded by a fluid space in the brain, and they can work as coaxial fluid-filled waveguides and resonance cavities, and because they also have red blood cells in them at various velocities, there can be some acoustic lens properties that are worthy of looking into. He mentioned the cerebrospinal fluid area, the ventricles and cisternal system, inner ear, and various air spaces in the head, all of which can be affected by directed energy, the modalities we're speaking about. The 9th slide shows you the resting state of a computational model that we've published. The reference is given below for looking at pressure wave applications and blast applications to the head. And if you shift to slide 10, you'll see the response of that, and particularly the pressure waves created in a simulation in a fairly detailed model in the cerebrospinal fluid compartment, the fluid surrounding the brain in the subarachnoid space. And notice how you see a spot down at the ear, sort of down, and then if you look up, you'll see a spot in this sort of cutaway view sitting over the temporal region. Save that image till we take a look back at some of the literature on exposures in the past. If we take a look now at number 11 in here in the series, you'll see this just another picture of the cochlea, and this shows you how the base of the cochlea, at the bottom it's the snail-like thing off to the right, labeled. If you look down and you see at the base, it's connected directly by fluid, by the endolym fluid, with the saccule and the utricle at the base. And the saccule and the utricle are shown at the upper left, and the semicircular canal crista is shown in that as well from just a histology picture of the human ear. They're sitting right out there. You can see the utricular macula is hanging out in space, and energy transmitted in can very easily move it. The next chart, number 12, shows you some pictures of the cochlea. And if you look down in the lower left, you see a schematic which shows you how when sound is incident, it causes something called the basilar membrane in the cochlea to vibrate at a characteristic frequency. This area has a resonant frequency distributed from base to apex. The base will move, especially as you move toward the high frequency end, so toward what we'll call the ultrasonic range. And up at the apex is low frequency, and that's shown here. It's transduced. The sound is transduced by hair cells, shown at the lower right, and transmitted via the vestibular nerve, via the eighth nerve, the vestibulocochlear nerve, into the brain. Now, if you take a look at the upper right image, A, you'll see something labeled the stria vascularis, which is a vascular structure that's bordering on part of the inner ear called the endolymph duct. And the next slide, 13, shows you a view of that. The stria vascularis is a vascular compartment involved in fluid exchange in the ear. And the important thing is, if you take a look at the picture on the left, that's an older drawing of it, and on the right is a scanning electron micrograph of a filled cast of it. You see that it looks like a whole bunch of blood vessels running through that area. Well, in fact, we know the dimensions. It has non-pulsable flow. It's tightly packed with blood cells through most of its area, and it's sort of the ideal area for showing both cavitation and acoustic lens type of properties that could aid in hearing. The slide 14 in here shows you that the phenomenon of acoustic cavitation. This is taken from a classic 1981 publication. In this case, the water was exposed to a 10 kilohertz signal, and you take a look, looking at the spectrum, you can see that there's a very characteristic power spectrum that's put out with increasing levels of 10 kilohertz ultrasound exposure into water. And so we can calculate, if we move ourselves up to slide 15, that basically if you take a look at the existing data, cavitation inside these systems are highly plausible. Slide 16 is something that I remembered from the earlier literature. It helps to have been around a few years. This is a picture these pictures show a side view. They cut the bone away from the side of the cochlea and looking at a guinea pig at the stria vascularis. Those are the vessels that you see, and it's filled with and there was a fluorescent marker put in so you could look at them with a video camera while you played 110 decibel broadband noise. And you'll see in the upper left all the vessels. If you now look at this upper right panel, you'll see that there are two spaces where there's a void in the vessel, not pinched off, but a void of some kind. And then when they turn the sound off, which is the lower image, the full vascular pattern was restored. What we think this is the sort of thing that one would see with dissolved nitrogen and oxygen gas, for example, forming bubbles under acoustic stimulation conditions, and adds to the plausibility of the type of a model that we were looking at. If you look at slide number 18, we move over toward another area in the literature, which are humans hearing microwaves. You're seeing this coming out in the press. Alan Frey is alive and well, who did this in fact, and he was just interviewed again. I spoke to him several weeks before about how they heard the radar emissions. He basically, someone said, "You can hear radar?" And he says, "Yeah, show me." And he went to a facility, and if we take a look at slide 19 coming up now, you can see the picture from one of the papers where he described this in 1963. They pointed the radar at him, and he did hear clicks. He held a piece of screening up on the side of his head, and the area where he could shield and not hear it is shown in stipple on his picture on the lower right side there on figure 3. And note how closely that lines up with the area where we see in the simulations acoustic-induced pressure coming up in variations coming up with ultrasound in the head model. Slide 20 shows us something even more interesting, though. This is work funded by the Office of Naval Research in the early 1970s, and these show you 3 recordings from the cochlea, from the round window at the cochlea of a guinea pig, exposed to pulses of 1 microsecond, 5 microsecond, to 10 microsecond duration radio frequency energy. And what you see the ringing that comes on afterwards. That's called the cochlear microphonic. It's at 50 kilohertz. It is in the ultrasound range for that animal, and this suggests that pulses and microwaves can actually induce a sound inside the ear through thermoelastic properties and maybe cavitation properties. So, in other words, the energy, the common feature, a common injury-inducing mechanism could be generation of sound inside the body by multiple forms of energy. The slide 21 cites a paper taken from the former Soviet Union where they showed that if you play pulses of microwaves at different frequencies to a human, they did effectively an audiogram, and we are maximally sensitive to 10 to 15 kilohertz pulse repetition stimulation. We hear it from microwaves. If you take a look at slide 22, this just shows, again, work supported by the Office of Naval Research. This was published in 1989, that in fact the auditory system and the brain responds to microwave pulses on cells that show regular auditory responses. And so what we come to, when we come to slide 23, this Frey effect, as it's been called, is a way that we can actually reconcile, and we can actually begin to view the notion that mixed kinds of energy, whether it's acoustic energy or radio frequency energy, or in fact we can speak about, because laser energy also can produce cavitation, that this could be a common mechanism for the susceptibility or vulnerability to various kinds of convergent directed energy weaponry. Next slide, if we take a look at 21, this just reminds us of the close proximity of the base of the cochlea, which is a place that rings when exposed to radio frequency pulses in animal experiments, to the saccular neutricles.